Welcome back to another episode of Day Zero to Breakthrough. I'm joining Elsha, a valued member of the team, as she continues progress on producing our vaccine candidates. Since we last checked in, the gene blocks arrived. Gene blocks are the pieces of DNA the team ordered from a company that synthesizes them. In our case, these gene blocks encode the Cyclone Alfred viral fusion protein. And the team has amplified and modified the gene sequences they need. They did this by using the PCR primers Jake and Nick designed on day one. Today, Elsha is extracting those DNA fragments, checking them for quantity and purity. These DNA fragments code for different versions of the Cyclone Alfred fusion protein and the clamp. The next step is to stitch these fragments together into a plasmid. Plasmids are circular pieces of DNA ordinarily used by bacteria to share genetic information with each other. Scientists use them to introduce new genes into different organisms, forming a construct which contains the complete genetic code for the vaccine candidates. This must be repeated 26 times, once for each vaccine candidate. I didn't realise quite how much work went into developing a vaccine. I was meant to join the team again the next day, but our plans ground to a halt when the university shut down for nearly a week due to the threat of Tropical Cyclone Alfred. So, I caught up with them again today. Since we last checked in, the team finished their vaccine constructs and put them into a bacteria called E. coli, which they used to produce lots of the DNA encoding their vaccine constructs. After a noble effort to keep cells alive and the wheels turning in the midst of wild weather, it was finally time to begin the process of producing protein. Varsha and Michaela suited up to transfect the constructs, putting them into Chinese Hamster Ovarian, or CHO cells. Transfection is the process of deliberately introducing DNA into a eukaryotic cell. We define a eukaryotic cell as one that has membrane-bound organelles, like a nucleus or mitochondria. This is important as using mammalian cells reduces the risk that the vaccine will produce an adverse immune response in their lab mice, and eventually, humans. The transfection must happen in the most sterile environment possible, as this drastically increases the safety of the process. It's a bit intimidating. This room is separated from the regular lab space by an airlock, is cleaned regularly, and the team are required to wear special protective equipment when they work there. Since this step was so important, I had to watch from outside. Luckily, Michaela generously agreed to get us some action from the inside. Varsha is an expert at performing these transfections, which involve mixing our construct DNA with a special lipid that is chemically similar to a mammalian cell membrane. Lipids are a group of chemicals known for not dissolving in water very well. You encounter lipids in your everyday life, things like fats, oils, and waxes. Lipids make up the cell membrane, which forms the outside of your cell and keeps all the important stuff on the inside. This is a very gentle method for introducing DNA that doesn't stress the CHO cells, increasing the chances of a successful transfection. Watching Varsha in her flow state, as Nick called it, was immensely satisfying. Every step was done with precision and care to ensure the highest quality. After doing this 52 times, twice for each vaccine candidate, the cells are placed on a shaker table to incubate, and Varsha got a well-deserved break. Over the next week, the team will carefully monitor their cells as they grow and produce protein. At the same time, they'll send the constructs off to be sequenced, which will help the team decide which proteins to purify. Next time we check in, the team will be beginning the purification process, which is significant progress towards the goal of vaccine breakthrough in less than 100 days. See you next time.